Hello, I am Charity Whitman. I'm doing the Lab 3 cheek cell DNA for Biology 2110. And this is my subject, my daughter, Carly. Say hi.
Hi.
I have already prepared all of our stuff. I have my lysis buffer right here, which is 1.5 milliliters of dishwashing detergent. I used Dawn, and I added 8.5 milliliters of distilled water and a quarter teaspoon of table salt. And the Protease solution I have right here. I measured out 10 milliliters of distilled water and put it in here. And I added an eighth of a teaspoon of meat tenderizer, and it's dissolved. We are going to go ahead and I already put the 3 milliliters of water, regular water, in here for her to swish with and spit back into that cup. Yep, go ahead. Don't swallow it, just swish it for 30 seconds. A little more power behind those swishes. I should have paused this. Okay, go ahead and spit it back in there. And now we are going to carefully pour it into here. I'm gonna put that back down there. Okay. I have this extra cup here to add in. And I have my clean plastic pipettes. And now we are going to add 2 milliliters of the lysis buffer to the tube of her spit, or her DNA. It's 2 milliliters. Yeah, I got a little too much in there. Let me do this so that. There we go. Okay, there's one. We get one more. That over there because it's dirty. And now we are going to gently invert the tube 5 times to lyse her cells. Which this is, I lost count already. Do it one more time. Okay. Do we notice any changes?
Maybe.
No. I don't notice any changes. It's just cloudy.
It's cloudy?
It just looks like the buffer solution. Okay, so now we are going to remove the proteins. Take this. Yes, we're going to remove the proteins. I'm going to take a clean pipette here and done with that. We are going to add 100 microliters, which on here is to the second line, which is 5 milliliters or 0.5 milliliters. This is the Protea solution. I hope I'm doing this right.
Hopefully.
That up there because I'm done with that one. I'm going to count this one this time. We're going to gently invert it. 1, 2, 3. Ew, I can see it already.
Oh!
4,
Oh my gosh.
5. It looks a little, just a little stringy. Just a little bit.
Strength.
Oh, I'm glad there was nothing in that. Can't tell you how many times we've done that already.
Wait, you did it more
Okay.
time?
Now we did that. We're going to put it in this cup and leave it upright, undisturbed, at room temperature for 10 minutes to allow the protease to work. So we'll be back. Alright, it's been 10 minutes. Now I have my isopropyl rubbing alcohol that I just got out of the freezer. And we are going to open that. Take this clean pipette and add up to 10 milliliters. Into this, holding this at a 45-degree angle. I'm going to do my best.
2.
Slowly and carefully fill the tube.
Why does it look like we're paused? Looks like you paused.
I'm not paused.
I know. Took us 8 minutes to pause it.
You can already see it separating.
I see nothing.
Hey, come here. Over here behind me and look at it. You can see the separation. Uh-huh.
That to that?
Yeah.
Oh yeah, like that's more clear.
Uh-huh.
Wait a second. Are you not supposed to mix it?
No.
That's cool.
You can clearly see the separation between the alcohol. And it's set up to 10 milliliters total of the alcohol. So I am going to do just that. cap back on. I can see a little bit right now. A little bit of stringiness.
Stringiness. Oh,
It looks weird.
very weird.
So I'm going to put it back in this cup
Sound.
and let it sit for 5 minutes and we'll be right back.
What?
Okay, now it's been 5 minutes
Yeah.
and it's almost her bedtime. We're going to see if we can see her DNA. I can already see little strands coming up. Stringy.
Stringy things.
We're gonna invert the tube 5 times slowly.
Excuse me, Missy.
Watch it, Carly.
I can see clear food. Careful. That's my DNA. Are you sure that's not food?
No, that's not your food. I can see the stringy white material. That would be her DNA. What do you think of that? Ew. Gross.
I never really liked it. Why do we need this?
Let's see if we can show it on her. I need a black background.
Hmm.
No, that's not working.
Like Jessie B or something.
No. It's hard to tell.
Oh, oh God.
What do you think about that? Quit trying to eat it. That's gross. Okay. That's her DNA. I'm going to properly dispose of that. And that's it for this vlog. Thank you for watching.
Bye.